Another day, another story. In December 2020, when pandemic lockdowns made in-person meetings impossible, hundreds of computational scientists gathered in front of their screens to watch a new era of science unfold. They were assembled for a conference, a friendly competition some of them had attended in person for almost 3 decades, where they could all get together and obsess over the same question. Known as the protein folding problem, it was simple to state: could they accurately predict the 3-dimensional shape of a protein molecule from the barest of information, its 1-dimensional molecular code? Proteins keep our cells and bodies alive and running. Welcome to Tabo Eminent Channel. Because the shape of a protein determines its behavior, successfully solving this problem would have profound implications for our understanding of diseases, production of new medicines, and insight into how life works. At the conference, held every other year, the scientists put their latest protein folding tools to the test. But a solution always loomed beyond reach. Some of them had spent their entire careers trying to get just incrementally better at such predictions. These competitions were marked by baby steps, and the researchers had little reason to think that 2020 would be any different. They were wrong about that. That week, a relative newcomer to the protein science community named John Jumper had presented a new artificial intelligence tool, AlphaFold 2, which had emerged from the offices of Google DeepMind, the tech company's artificial intelligence arm in London. Over Zoom, he presented data showing that AlphaFold 2's predictive models of 3D protein structures were over 90% accurate, 5 times better than those of its closest competitor. In an instant, the protein folding problem had gone from impossible to painless. The success of artificial intelligence where the human mind had floundered rocked the community of biologists. "I was in shock," said Mohammed Al-Quraishi, a systems biologist at Columbia University's program for mathematical genomics, who attended the meeting. A lot of people were in denial. But in the conference's concluding remarks, its organizer John Molt left little room for doubt. AlphaFold 2 had largely solved the protein folding problem and shifted protein science forever. Sitting in front of a bookshelf in his home office in a black turtleneck, clicking through his slides on Zoom, Molt spoke in tones that were excited but also ominous. "This is not an end but a beginning," he said. "Share this article copied. Newsletter Get Quanta Magazine delivered to your inbox. Subscribe now." Recent newsletters. Proteins are molecules that come in hundreds of millions of different shapes. Each one serves a particular biological function, from carrying oxygen through the blood to sparking chemical reactions. The function is typically defined by its shape or structure. Quanta Magazine. Source: RCSB PDB. Introduction: when Google's public relations machine churned the news out to the world, the media went wild. Headlines claimed that AlphaFold 2 will change everything. Protein biologists who had spent their entire careers investigating the structures of single proteins feared that they would lose their jobs. Some claimed that AlphaFold 2 would revolutionize drug development. Now that biologists could quickly learn proteins' shapes, they could create new medicines that could target them. Others pushed back, arguing that the results were mostly hype and little would change. Molt could barely comprehend the news himself. He ended the conference with the question on everyone's mind: what now? That was 3.5 years ago. It's finally possible to start answering his question. It's the biggest machine learning in science story that there has been. Mohammed Al-Quraishi, Columbia University. AlphaFold 2 has undeniably shifted the way biologists study proteins. However, while AlphaFold 2 is a powerful prediction tool, it's not an omniscient machine. It has solved one part of the protein folding problem very cleverly, but not the way a scientist would. It has not replaced biological experiments but rather emphasized the need for them. Perhaps AlphaFold 2's biggest impact has been drawing biologists' attention to the power of artificial intelligence. It has already inspired new algorithms, including ones that design new proteins not found in nature, new biotech companies, and new ways to practice science. And its successor, AlphaFold 3, which was announced in May 2024, has moved to the next phase of biological prediction by modeling the structures of proteins in combination with other molecules like DNA or RNA. It's the biggest machine learning in science story that there has been, Al-Quraishi said. However, there are still massive gaps that artificial intelligence hasn't filled. These tools can't simulate how proteins change through time or model them in the context in which they exist, within cells. AlphaFold changed everything and nothing, said Paul Adams, a structural biologist who develops algorithms to model the structures of biomolecules at Lawrence Berkeley National Laboratory. This is the story of how Jumper's team at Google DeepMind pulled off their coup in protein science, and what it means for the future of artificial intelligence in biology. Formulating the problem, a piece of origami paper is little more than pressed wood pulp until it's folded in specific ways. Then it becomes something new. A few precise crimps and flips, and it's a fortune teller, a paper device that can predict your future. Take the same piece of paper, change a few of the folding steps, and now it's a winged crane, granting good fortune to its recipient. Similarly, a long string of amino acid molecules has no function until it spontaneously folds into its innate shape, which biologists call its structure. A protein's structure determines how it binds to or otherwise interacts with other molecules, and therefore defines its role in a cell. There are a couple 100 million known proteins on the planet and many more unknown ones. They do it all. Hemoglobin and myoglobin ferry oxygen around the muscles and body. Keratin gives structure to hair, nails and skin. Insulin enables glucose to move into cells to be converted into energy. Proteins can take on a seemingly infinite number of shapes to match the seemingly infinite number of jobs they do in life. Right from the atom all the way to ecosystems, protein structure is kind of a lingua franca, Al-Quraishi said. It's where everything happens. A cell makes proteins by daisy-chaining small molecules called amino acids into long polypeptide strings. The amino acids it chooses depends on the underlying set of instructions provided to it by DNA. Within a fraction of a second of its creation, a polypeptide string bends, buckles and folds precisely into the protein's final three-dimensional shape. Once off the molecular assembly line, it scurries along to do its biological work. If proteins didn't perform this folding process exceedingly well, cascades of disasters would tumble through the body. An incorrectly folded or unraveled protein can lead to toxicity and cell death. Many diseases and disorders, such as sickle cell anemia, are caused by misfolded proteins. And misfolded proteins can aggregate into clumps that are hallmarks of neurodegenerative diseases like Alzheimer's and Parkinson's. Yet no one really knows specifically how protein folding happens. How does the sequence information in these simple molecular chains encode a protein's complex shape? This is the most profound question that we can ask, said George Rose, a biophysics professor emeritus at Johns Hopkins University. Thanks for watching. Request you to subscribe the channel.